I'm very happy to introduce another member of our Scientific Advisory Committee, Ernesto Lupercio, who was one of the original members of our Scientific Advisory Committee, who to introduce the next speaker.
So many members of the advisory committee. It is a delightful pleasure to introduce Dr. G.Olmedo. She's my colleague at Simvestab. Simvestab is meant to be the elite research institute in Mexico that is not quite associated to a university, a little bit like the IAS in the US, and the fields in which it is strongest is actually the biological sciences. Dr. Olmedo is one of the finest biological minds in Latin America. She is a researcher at the Department of Genetic Engineering at Simvestab and the director of the Irapuato campus of Simvestab since 2013. She got a PhD in macrobiology from UPenn in 1990 with the advisor Philip Youngman, and she has been a visiting professor at the Salk Institute. Her fields of expertise are RNA biology and more recently molecular ecology. She has investigated with her team made amazing discoveries in the molecular ecology of the Cuatro Ciénegas ecosystem, but we may hear more from her in her talk. So it's a pleasure to introduce Dr. Gabriela Olmedo.
So thank you, and I want to thank you, Stephen, for this invitation, also for the pleasure of being part of the advisory committee to the Institute of Mathematical Sciences of the Americas. And I'm really glad to be here in the company of Mina for this particular public talk. And just to start, I'll tell you that my work has to do with bacteria, with microbial communities. Now let me tell you some of these properties or the things that we don't know. We figure these are sort of like networks, and we wonder, how does the network work as a whole? How do they speak to each other? What are the rules? Is there a backup system? Which is the language mechanism? And how to handle huge amounts of data? So if you remember, this is exactly what Mina was telling us. These are exactly the same problems that people face when they try to understand how neurons work. And that's a little bit of what happens in my work. So in my work, say like the magic of my laboratory is the experiments. We are very good experimenters. So I can pretty much make things work in the lab, in an amazing way. But we really need the bioinformatics, the computer people, the mathematics, and the physics from other people to not just understand our data sometimes, but to make models and hypotheses, to then go back to the experiments and work on this. So we really, work very close to a lot of people so that we can, do this work. And this is what I'll tell you about, today. I will, give you a primer on microbiology. I'll tell you that the DNA is at the origin of evolution of life. And this is the main questions. How can thousands of species co-occur in a community? And these communities, you'll see that you can, think about that of your brain, you can think of that of your gut, you can think of a forest or anything that makes a community. And then I'll tell you, that we want to understand what are the rules of assembly of communities. I'll tell you a little bit how we set up these things in Cuatro Ciénegas, which is an amazing place in Mexico. And finally, how we try to reduce all this information into more simple systems in synthetic ecology, where we try to understand only a few cells at a time, and we always are blocked or stopped by emergent properties. So we'll start with bacteria. And bacteria are these small things that we do not see, but that they can divide very quickly. Maybe every 20 minutes you will have a clone of a bacteria. So from one bacteria you get 2, from 2 you get 4, 8, 16, etc. So you immediately see this exponential pattern, okay? So this is, an interesting pattern in which bacteria are just duplicating all the time. And a lot of people say, okay, so what is the limit? I mean, can they cover the whole earth? Of course they cover the whole earth. They can inhabit every niche, but not numerically, because they have a limited. It happens that after some time, bacteria find themselves into an ecological problem. So ecology is the limit for bacteria, and that's why they don't just continue, growing and growing all the time. So if you actually graph bacterial growth, you'll find more like an S shape. And this is because between the R, which is the velocity of growth of an individual, and the N, which is the number of individuals, a K gets inside, which is something that limits the population size per capita. So K means what is the maximum real value that any species can get? How many of them can we get? So if we start with a very small n, with a very few individuals, and a high capacity for growth, we pretty much go to an exponential phase, which is here. But if the number of people, bacteria or whatever gets larger, then you start to fill up all this capacity. And then really growth has to stop. So what is limiting this? It's a limitation for resources and it's competition. So this is how I do my work. I use Petri dishes. And so I don't have a forest, I don't have to work with electrodes and things. I have my Petri dishes. Bacteria don't complain, okay? I can just have thousands. So I have all these bacteria. I only really see them with an electron microscope or another optical microscope. But I can just use a Petri dish, and then every one of these little dots, it's really a colony. A colony forms when a single cell lands on a Petri dish that has nutritious media. And then in just 18 hours, I can have 10 to the 7 or 10 to the 9 cells, okay? I told you, exponential, growth. So next day, I can readily, my students can set up an experiment. Next day we have results. We have this Petri dish, and we have all these bacteria. However, you can see that some bacteria are very large, some are small. What is happening is that's what we're seeing. We are seeing a resource restriction. They have a different K, different, R, and so each one is, really playing by different rules. But when they get together, then they sort of are in each one's way. And so what happens is, for instance, this one cannot reach more because it just reached out of space, right? The Petri dish ended. And some of them, like this one here, maybe some should be just as large as these ones. But if they are very close to the large ones got the resources, okay? So you can do ecology just in a simple Petri dish, and that's, a lot of what I do. But one of the questions is, where do bacteria come from? I mean, why are there so many, and how are they distributed? Why do I have some in a Petri dish? So there is this idea in biology that everything is everywhere and the environment selects. So we have this idea that bacteria and in general microbes are very good to disperse in the world. I mean, they are so good to move because they can move with water, they can move with wind, and we can move them around when we travel, right? Like the coronavirus, and that's how we move things around. And everything moves around because it has no restriction. If you don't often see birds, or some things out of place. Like bears, they are restricted by the food they eat, right? They are restricted by the environment they need. But what is the restriction for bacteria? So that's the question here, is like, if bacteria can move around everywhere, do we find the same bacteria everywhere?
Even outside Earth.
I know. It's a real concern. If spacecrafts are taking the spores of bacteria out to other places, okay, so they really decontaminate, the spaceship all the time. And there is also a question: did spores actually, came to Earth? Is that the origin of life on Earth? Right? They just came because they are very resistant.
You see, coronavirus.
Excuse me?
Coronavirus, yeah.
Coronavirus.
It's not bacteria.
No, it's not as resistant. Coronavirus would not be a problem. You all know anthrax. Anthrax is a problem because it makes spores. Spores can last 100 years. In some experiments in Frederick at NIH, they had to close a building where they were working with anthrax because it was easier to close the building than to clean it. Okay, so some things are more difficult than others. So we are sort of like an example of a community of bacteria. We have so many cells inside us, and we have even more bacteria, 3 times more bacteria than our own cells. So now we wonder if our genome, that's all the genes that we are more bacteria than a eukaryotic cell. Right? Because we have many more genes and many more things because of the cells that live with us. And so the question is, how do they get there? How do even we measure this diversity? I told you, we can see it on a Petri dish. But I didn't tell you that my Petri dishes can only cultivate 1% of what's outside. My Petri dishes are not telling me the truth. They are just letting some things grow and not others. Okay? So how do we actually know what lives, in us, in human cells? And then if we know that bacteria go everywhere, you could have a first hypothesis that if I take a sample of different places in a human being, and then I do the same experiment a few weeks later, maybe because this human being just washed their hands and did a lot of things, maybe this microbiome is going to change, but it doesn't. The thing is that once a community of bacteria establishes in your body, in any part of your body, it's really there for a while. It's very stable. And the same is true for the bacteria that you will find in soil or that you will find.
Our cells are changing, the bacteria remain?
The bacteria remain. Yeah, you can kill them if you take an antibiotic, for instance, and then you make a mess of
But
a
you
lot
talk
of
about
things.
skin bacteria, but the skin is changing every couple of
Oh
months.
yeah, but the
right?
bacteria, they remain the same. Yeah.
Well, in the same tone though, the bacterial cells also are new cells, insofar as they're multiplying and dying off and new cells being replaced.
And basically, bacteria are using our lotion, and they are using our cells, for growth. So pretty much different races, different at different ages of your life, you will be changing also bacteria, depending on hormones and other things. Yeah. So that was one sort of like surprise. I mean, bacteria are everywhere, but it's not like they just spread and you just get a new collection every time, right? It's like you really have a stable, community every time. So one other question is, how come there are so many different bacteria? Because the diversity, as you will see later, is amazing. So everything in life comes from the same molecule, which is DNA, okay? In DNA, this is the chemistry of this, it's got sugar and phosphorus, and it has some, 4 bases like A, C, G, and T. And just the combination of these 4 letters in different lengths and settings can give us all the diversity that we have in life, okay? So in bioinformatics, that's all we need to know. We need to know that, this combination and what's this for, and we have bioinformatics. So let's make a test now, okay? You are the bioinformaticians, and I have a strain. This strain has this combination of bases in a particular gene that we use as an ID. So what strain is this? This is your database.
What's the
Acetobacter. Yes? So this is your strain 1, GGAACZ. There you go, it has this, it matches this. So it's an Acetobacter, okay? Have strain 2, you know, the same thing, you check this out, check your database, and you cannot read it, but it's Campylobacter, okay? So the other one is easy because you just deduce that it's E. coli, but yes, you can check it out, TTC. So this is bioinformatics, okay? And that's how we know what things live in this planet.
This is only for the ribosome?
This is just for the ribosome, yes, yes. Now it's getting more complex because we can get all the DNA. But we really need a tiny bit of a piece of DNA from the same region every time to compare. We need databases, and that's it. What else do we do with these pieces of DNA? We can align them together. We can put them together and then see how much they look like the one below. And every time there is a difference, I'll put it in green. See, so here you have a G, but then you have a T and A and other letters. And here you have C and T, etc. So what you can do now is that you can cluster these in a way that the ones that resemble the most are closest, and the ones that are more different, are separated. And then you can construct these sort of cladograms, okay, that we can call them also trees. And that's how you can know how evolutionarily close some things are or how different they are. You can calculate how many green things do we need to get a new species, okay? And so we can classify everything that is living, and that has DNA, and we can classify these and define where they are in these trees. So we can know how far we are from fungi. We are here in the Metazoan, and we are with the Drosophila and with all the things and vertebrates, and you won't believe that we are just this tiny thing. You're here, okay, with the vertebrates, okay? You can do these for everything. This is done for trees, for instance, and with trees, you can also classify trees, same trick, just put it together, do this, and you will know what you have. And then if you want to understand what life really is, just put it all together. Put bacteria, put fungi, everything that you know, that you've heard of, just put it here. You can put here even things that are extinct, because you can get that DNA and put it in this tree. And what do you find in this tree? All of these are bacteria, okay? So it's all bacteria, just don't bother, don't worry about anything else, because all of life is still bacteria, okay? If you go into this branch, then you find eukaryotes, and then you'll find this little tiny thing, which is the opisthocomta, that has the metazoans. And even below this green is us, okay? We didn't even feel in these, okay? So we are nothing. We're just a little dot here compared to what's really in life, what's maintaining life together. Because it is bacteria that actually are doing the cycling of all the nutrients in life, in the oceans, the carbon. They are feeding the plants, they are feeding everything, okay? So yeah, I'm a fan of bacteria. But how do you actually go and if I cannot even take out a bacteria, how do I even know that it exists? So I'll show you what you do when you cannot count one by one, okay? These scientists took a car and then just drove, I don't know where in the US, and they just didn't, clean the whatever, what's the
Windshield.
Okay, they didn't clean it, and then at the end of the trip, they collect everything. They use some alcohol, and then you take out all the DNA there, and then you sequence that, and then you go to the database and say, what are the insects that live in this area? Okay, there you go. You have the information, and this is what we do with bacteria. You can go and take a swipe, of some part of your skin or anything, and then just use alcohol, DNA, and get the same information. So this technique of metagenomics really changed, the way we think about biology. It really sort of like, woke up microbiology. Because then we realized how many bacteria and what was the diversity of bacteria in the world. And that's where Cuatro Ciénegas comes in this plane. So Cuatro Ciénegas, it's a place in Mexico at the north. This is part of a second largest desert in North America. So we have part of that in the south of the United States and Mexico. We share this desert here. Coahuila is here. I live in Irapuato. Lupercio lives in Mexico City. Some people know Cancun, okay? And you find Florida here, okay? So this is easy, okay? Cuatro Ciénegas is there, but where was it 100 million years ago? 100 million years ago, Coahuila, is nowhere to be found here because it's right here. Cuatro Ciénegas is actually part of the Coahuila, and Coahuila had a shore, you didn't have to go to Cancun, you could go to Coahuila, but then the Gulf of Mexico emerged, and then no more ocean, but the water was still hidden below, the mountains, and it makes the ponds, and it's still there. So this water is actually, creating an oasis of bacteria that are really amazing and sort of like remind us of Jurassic Park, because you go there and you find everything that was in Jurassic Park, okay? So this interior basin has a lot of springs, it has many pools, it has many endemic fishes, and it has some weird properties because it has so little phosphorus, so little nutrients, that's what's kept this place alive, that it's so poor in nutrients that not a lot of things can live there. So this is a pond in Cuatro Ciénegas. When you see a pond that is very clean, it means that you don't have nutrients, okay? It's very poor. It doesn't have a lot of nitrogen, it doesn't have a lot of phosphorus, so very few things can grow. But one amazing thing is that you have these kind of stones that are not stones. These stones are stromatolites. So stromatolites are actually bacteria. These bacteria is sort of like they grow together and they have layers, and then a lot of carbonates sort of like precipitate on the slimy surface, and they actually become stones.
These are dead bacteria, right?
No, that's the important thing. In Cuatro Ciénegas, these are alive. So you have a lot of tiny things still making their way, and they're still growing. You find the stromatolites, lots of dead ones in a lot of places, and those are the fossils of bacteria. I mean, it's very hard to find a fossil of a bacteria, come on, right? Sure. But if you find a stone from a stromatolite, then you know that was, bacteria were there. So that's what we know about stromatolites. Those are the fossils of bacteria. Those are the keys to tell us what was the origin of life on Earth. So the origin of life, I don't know what happened before because we don't have the stromatolites. But we know how old stromatolites are. And so we know that in Cuatro Ciénegas we have a very special place where the stromatolites are still alive. We have the same types of bacteria, that live in other places. And then also we have, an idea of an old chemistry, okay? Because when the planet was forming, of course, the atmosphere was not with oxygen. So what happens is that it took a long time before oxygen started to appear there. It required for the evolution of cyanobacteria here, which are photosynthetic bacteria. And these bacteria started to use CO2, make oxygen. Oxygen, of course, is very toxic. No one wants to live with oxygen if they are anaerobes. So the chemistry of the whole planet had to change. Because there was no oxygen, even the phosphorus was not linked to an oxygen, it was linked to a carbon. That's phosphonate, that's what Monsanto used for herbicides, okay? Because no one can grow with this thing, okay? But later in evolution, then in an oxygenic place, then you have now oxygen and phosphorus. Other things that were not common, like copper, you didn't see them free here. Now they are free. So even copper sort of like starts to get used in evolution. So just to tell you that the chemistry of the world is really linked to the evolution of things in the planet. Some organisms used hemoglobin to make their blood and to circulate oxygen because they used iron in this molecule. And other organisms like squids and octopuses, they chose copper to move oxygen, okay? So they have hemocyanin and those are blue. So some organisms have blue blood and some have, it's not really blood, but okay, that's what it is. And so that tells you how critical it is when life starts and evolves. What is the chemistry? I mean, the organisms are changing the environment, but the environment is also changing the organisms, okay? And these stromatolites, if we open them up, what we find is that we have different layers. Each of these layers is composed of different organisms that have different strategies in chemistry. So you have some that prefer sulfur, and then life starts evolving from no oxygen and sulfur to oxygenic, and then now you use light. So this is the evolution of life. So if you take apart a stromatolite, you have the record of life there, of how things evolved, okay? So that's what it's really fascinating to have in Cuatro Ciénegas. If we do a metagenome, sort of like the windshield experiment, but in the Cuatro Ciénegas, in one of these places, which is called Churince, this is the bacteria that you find. Each of these things means to be, a phylum of bacteria here, okay? So you find 66% of the bacteria known in the world, you can find them in Cuatro Ciénegas, okay? And I'll tell you something about this particular group, which is the one that I work with. If you take that bacteria and then just take those that you can culture, we have, cultivated in the lab, more than 1,000 strains just of bacillus. And then if we ask, how many new species do you find? All the blue are the new species that we find, okay? This is like the world of 100 years of people looking for bacillus. After 2 years just looking in Cuatro Ciénegas, we have 26% of the species, that were not previously found in the world, and now they are found in Cuatro Ciénegas. So, we have published this, and we call Cuatro Ciénegas the lost world, because we're really finding a lot of organisms that are not found in other places in the world, that we think are part of the bacteria that were, still in this, underwater that was previously an ocean, okay? In fact, many of those, when you culture them, they look very much like some things from the oceans. Okay. But what my lab really wants to understand is all these bacteria, how come they are, together forming communities? So if this is a community and each of these, dots with colors is a species or a bacteria or, any strain, how come they are together? So one is a stochastic model in which, okay, you just are born there, or some were extinct, or some came from dispersion, and they were randomly selected from the environment, okay? So how different would this be from a car community, in a parking lot, okay? Would be the same thing, you would just toss them around and you will just find them randomly? Or is there anything else? So that's sort of like the null model, okay? The null model is you just toss around bacteria and you find communities, and it doesn't mean anything because it's all stochastic. But then you can also think about a deterministic model in which they each have a different function, and not just that, they help each other to grow. And so if they are interacting, if they are cooperating, they are competing, then it's not stochastic. Let's look at the DNA composition of 3 bacteria, 3 green bacteria that you find there. So what I have here is that this is the genome of one bacteria. These are the composition of its genes. This is another one. This is another one. All bacteria share a group of genes that are needed to live, not to grow, to reproduce. They all share those. But then bacteria also have specialized genes that you have their viruses, transposons, you have sugar utilization things, phosphorus, antibiotics, synthesis. So you have a lot of things that represent the ecology of the place. So bacteria have to collect a set of genes to be able to live in your gut or to be able to live in Cuatro Ciénegas, okay? If they don't have the right genes, they cannot colonize your gut because they will not be resisting the pH, coming through, or the anaerobic conditions. So it is a set of genes that allows bacteria to actually colonize something. And so we think then that some of this is deterministic. If we take a collection of bacteria, again, this is a tree, and this is how they are, the different groups. Each of these dots, if you see it in a color, means that it has that function. If it's got no color, it lacks the function. So what we found here is that a lot of bacteria are really mutants. They are lacking, part of things that others have, okay? So it's like you get to the exam, I have a pen, you have paper, okay, let's split the pencil, give me a piece of sheet. I mean, so you can, you have to share resources. So this is what we have. We have that bacteria really have incomplete genomes, okay? They don't have enough genes to get by in an environment. They really need to sort of like share things with other bacteria. For instance, this is a petri dish, and you don't see anything because nothing grew. And you don't see anything because it just also did not grow. But if you get these bacteria together, then they grow. Because this bacteria, the 316, is feeding something to this other bacteria, and this bacteria is maybe feeding something else to the next one. So this is what happens in communities, that bacteria are actually sharing information, they are sharing genes, and that is what makes things possible. This is a model where enzymes and genes are public goods, okay? So you don't have to have a full collection of genes to live in a given place, because you can have 10% as long as your neighbor has another, other ones that you need. So you can really be sharing all these public goods. For instance, for phosphorus, you don't need to be able to use all the sources of phosphorus. If some use DNA as a source of phosphorus, if some use calcium, phosphide, whatever, at the end, all of them are going to become, inorganic phosphate, and anyone can use it. So that's what these public goods are, okay? And that's what makes it, it's sort of like layers of complexity and complexity and complexity as you go deeper in understanding communities. So I wanted to understand a particular community in this place, which is the Churince. So I take these very tiny samples, and I ask, do these bacteria grow together or they kill each other? What are they doing? So we actually asked whether they could produce antibiotics, which is a weapon in nature, right? Bacteria produce antibiotics and kill each other. So what you have here on the background is a bacteria growing. And on the top, you have other bacteria growing. And then this sort of like halo that you see, it's because bacteria were not able to grow there, okay? This one is producing an antibiotic that it's not allowing growth of this other one. So we can test this for 78 strains, okay? So we do a matrix of 78 times 78 times all of the reproducing experiments that biologists need, okay? And we can have a network, and we can ask, how do these bacteria get together, okay? Do they get along well? Each of these circles represents a bacterial strain. The green bacteria do not do anything, they are just there, okay? But this bacteria here that is connected here, okay, with this graph, this connection means that this is killing everything that is connected there, okay? So it's really a carrier, it kills all of this. And then you have all of these bacteria that are being killed by somebody here, okay? So we did that for a lot of bacteria, so we know which bacteria don't seem to interact, which bacteria are killing others, and which bacteria are sensitive. Then we gave up because it was too much work, okay? And then we decided to do something more simple. Say, why do we have to understand all 78? Maybe we can see what's happening with 3 of them. So we took 3 of them, one from the top that did not kill anybody, one that killed other bacteria, and that one that was sensitive. So we have this community, we have the data for each, we constructed a matrix here, and we have the ID for each of the 78 bacteria, and we know who they kill and who they don't kill. So we can do then this cellular automaton to figure out whether, I mean, why are these bacteria there? Why aren't these dominant bacteria that make antibiotics, why aren't those the dominant thing and the other things just are extinct? I mean, so in this cellular automaton, we seed, these strains, okay? Every time a strain grows, if it finds a sensitive bacteria, it's going to kill it. If it finds, resistant bacteria, it's not going to kill it. So at the end, what you have is this. There's no extinction of these bacteria. In fact, you have sort of like patches, and these patches really have, like these bacteria that are killed often, as long as they are surrounded by resistant bacteria, they will not be antagonized, okay? If you repeat this experiment, but now you shake, your in silico plate so that you get a lot of perturbations, then when you have this extinction, then immediately some of the bacteria, the sensitive ones, are going to die, and you will only have the resistant bacteria and the antagonistic bacteria, okay? The sensitive are gone. Can we do the same but with real experiments? So we took one bacteria of each, okay? We can tell them apart because this one makes this kind of colony, which is larger and sort of like a cream color. This one is a bit smaller, and the sensitive is yellowish. So we're going to do the same experiments, okay? We are going to put together the antagonist and the sensitive, and then we're going to put them together, and after 5 minutes or 30 minutes, we'll see who survived. Then we'll do the same, but with all 3. After different times, we'll see who survives. So what happens if I put together the antagonist and the sensitive? I know that this one kills this one. Well, the antagonist, nothing happens. After 30 minutes, it's still there, okay? I'm measuring growth in a petri dish, okay? And this is time. But I can see that the sensitive bacteria died, okay? You'll see that it doesn't really die completely. Some of it is still there, but I'll tell you more about that later. So what can we say? Okay, this is easy, right? I have the antagonist, and it grows and the sensitive declines, so there is a competitive interaction. If I have a sensitive and a resistant, nothing happens. Both grow, nothing happens, no interaction. If both grow, the resistant and the antagonist, they don't do anything to each other, they grow. So what's going to happen if we just grow the 3 together? So that's what happens. What happens is that when we grow the 3 together, there is no more killing, okay? The sensitive bacteria survives. So even in a few minutes, there's no killing, so this is an emergent property. You couldn't have expected this to happen. And it really behaves in a petri dish just like the cellular automaton. You have that some of these bacteria sort of like find a little patch far away from the antagonist, and they survive. If they have the large ones, which are the resistant, they also find a place surrounded by these resistant, and they survive, okay? So So we do all kinds of things to understand what's going on, that now we can put some inside a membrane and the other one outside, and we want to figure out, if is there an antibiotic that is killing it or not. So what we find is the same thing. Even if we put them apart, they don't have to touch each other. It can kill it even when it's one in the dialysis bag. But this is what I wanted to show you. It doesn't kill all the population. There's always something remaining. What is that thing that remains? Is it like the population has sensitive, but there are always survivors? This is a very important question because this is what we find in medicine for antibiotics, okay? We cannot always kill everything, and that's a big problem for the systems, okay? So well, again, you need a dialysis bag. If you put the 3, they all survive. So what happens if I take some of the surviving bacteria, okay, and if something happens there, they are not dying anymore. So I'll put fresh bacteria here and see what happens. If I add fresh of the sensitive bacteria to this flask, it doesn't die. Something changed in the media, something changed there. I have no idea. It's completely unexpected. Again, it's something you cannot predict. This is another experiment. I take some of the bacteria that already was in the presence of the antagonist, and I put it with fresh antagonists. Say, let's see if now it's going to be killed. And what happens? It survives. Otherwise, I wouldn't be telling you all of this. So again, we have these emergent properties. And why is this important? Because this was for the model. The model is, this is so easy. I have a resistant, antagonist, sensitive. But the real thing is that this one is changing things for this one. This one is changing this for this one. It's also this one, to this. So there are all these interactions going on here, okay? So even with 3 bacteria, we have no clue of what is going on. I think I have 5 more minutes. Yes? Okay. So that is an interesting question now, and it's sort of like if you look at the literature these last 2 years, everything is about higher order interactions. And higher order interactions means that sort of like the classic ecology, Lotka Volterra, which means it's like as long as you have a lot of rabbits, then the population of the predator is going to come up. Once the population of the predator comes up, then the rabbits are going to go down. And this is a classic equation. But now that in communities we have thousands of bacteria and we have emergent properties, it so happens that people are trying to find, sort of like to do something to Lotka Volterra to be able to do something, okay? So we have these analyses, these suggest that we have a higher order interactions. We're doing the molecular biology, we're doing other analyses. But the truth is that it's sort of like the place where all the ecologists are trying to understand what is going on in nature in any community. So if you have a community like this one, you have a shrub, and in the presence of the shrub, sort of like the grass grows better, and that's because this one is limiting growth of this, so that leaves more room for this one. So the thing is, things that you cannot predict because there's just not an interaction of 1-to-1. So this is, what's going on now in biology, that you don't know how to predict what are the stable combinations in anything. And I don't know if you ever take probiotics. Okay? Who knows what we're doing with probiotics, okay? We don't know what we're doing. In agriculture, we are putting in a lot of biofertilizers, which are bacteria and fungi. We don't know what's going on with any of these bacteria or fungi there, okay? So this becomes a very important thing because when we take antibiotics, we kill a lot of bacteria, but not all. So what happens with the community that remains? It's really a problem. So the thing is, we don't know anything, and there are plenty of papers that do a lot of mathematical simulations. tell me any model that you want in mathematics and economy, and it's been tested now on community ecology, okay? If you want to know, for instance, in the gut, what happens with a probiotic species, when you know, just let me know. How often can I find two bacteria? How often do I find 4? How often 6, 8, 10, 12, 15, 20, okay? And it goes on, and I cannot predict much, okay? And what happens is that the assembly of communities as they grow makes prediction, you know, much more difficult, okay? So this is one of the main problems now in studying, you know, these communities. And this is just looking at species. But what about looking at the genes there, okay? So that's another layer of complexity, okay? So that's the same thing for insects. If you want to predict for bees, and you only have 10 different species or 10 taxa, and you want to know how many combinations you can do, and so that each insect is going to carry inside any of a different combination of 10 taxa, and then you ask how many of those combinations are going to be stable. So that's why you have like different colors, which tells you how many times the same combination, you know, survived. Okay. Again, this is very important because there's a lot of mortality in insects and bees, and everything has bacteria, and everything has, you know, that is alive has bacteria, so it becomes a very important work, okay? So we'll continue exploring functional traits, you know, the competition, we'll figure out if we can, you know, simulate invasions and see what's going on. And then this work in Cuatro Ciénegas is also serving the purpose of, you know, making aware of, you know, to the population that you have to be careful with water, that you cannot cultivate alfalfa in the desert because this happens, okay? So they cultivate alfalfa in the desert in Cuatro Ciénegas, and some of the ponds are drying, okay? So it's like, yeah, evolution made all these efforts to maintain this Jurassic Park, and it's gone, okay? So there are other surprises in Cuatro Ciénegas. We are working with other things that seem to be, you know, even older. And, you know, so we still have a lot of things to do, but we are worried about Cuatro Ciénegas. We run molecular biology laboratories. We have in the high school in Cuatro Ciénegas a molecular biology lab. So we work with high school kids in there so that they sort of like understand the science with the scientists, know what we are doing. They work with us. And we also run other workshops, you know, to attract, you know, young people to science, to this kind of work and experiments. And this is my lab, okay? And my collaborators, we only drink beers, and these guys do the work, so it's great life, okay? And of course, we have to find money, and so we usually have money from CONACYT, which is the research, you know, foundation for funding science in Mexico. And well, we also got money from part of this from the World Wildlife Alianza Carlos Slim, and so that's how we sort of put together this work. Thank you.
Question.
When you mentioned the 3 species that when you added the 3rd one, the weak one survived, I just wonder, it's just a thought, if maybe there's some model of game theory that applies there.
What model?
Maybe there's a model of game theory that applies there.
Yes, yeah. There's, I don't know if you probably know better. I'll give you the papers. They have done every model for different communities. The only problem that I see in these papers is that they just look at, you know, at the presence, absence of different bacteria or different things. It's like they take a sample with millions of species and they ask, do they co-occur? You know, do I find the same 2, the same 3, the same 4? That there are no other experiments. I have a collaborator, he's doing this marriage problem in which he wonders what kinds of sugars or preferences, you know, in substrates they are using, and to try to find something, you know, behind that. not just a model, but some experiments to see their preferences. You know, if you, in this, you know, game theory, what would be your preferred, you know, sugar or substrate? And, you know, would you be taking, you know, from your partner or not? But so far, there are no experiments. These are just models. You know.
And the second question, is your work influenced by the melting icebergs, by climate change?
No. I do have another work that has to do with experimental evolution to temperature.
So when you have the sensitive bacteria die off and there are few survivors, are those survivors genetically distinct from the ones that died?
Okay, we have not sequenced them. I mean, we don't know all the detail. We just know that we can recover them and put them again to test, and then they behave identical to before. They will also, half of it, they will survive. So, I mean, they are not changing what they do, but I cannot
So
know.
there must be some different properties.
Evolution happens so quickly in bacteria. We have mutations so quickly, but I don't think we have 20% of the population mutated so quickly. So I think that it's more a regulation thing. Maybe there are genes that are turned on and off, and you can do that very quickly in 20% of the population.
But in fact, I was thinking along the lines of what Jose just said, which is when you re-inoculated, did you inoculate from the residual population or
Yes.
from your initial inoculum? Because I think the answer is inside there, because if you're re-inoculating from your residual population, then in fact you've enriched for.
Oh yeah, but that's the idea. I mean, we take the
Right. So
survivors, so we
therefore-
know we enrich that, and we know that's what are left. So we do both things. We start fresh with a new population, and we take the residual population, which we know is just the enriched survivors. Yes. So we do both.
I have a very question. You show numbers several times. For example, 66.28% of biodiversity was concentrated on certain single lake. When you say 66.28%, do you mean by head count of species, or do you mean by furthest genetic distance on classification tree, or maybe this is pairwise morphological accumulation?
It's, you know, it's of all phyla known in the world, we can find 67% of those in this particular place. It's just a percent of you know,
The species.
of the species
Of the-
at the phylum level.
Yeah.
Okay, yeah.
I have a question, different topic. You used the word structure. So does that mean potentially physical structures
Yes.
where either nutrients, novel metabolites, for example, could actually be concentrated? So if the same experiment is run on a solid substrate, like a plate, as opposed to a broth, would you get different survival?
Absolutely, it's completely different. And structure does mean that. So most bacteria actually live in communities, in biofilms.
Yeah.
So these biofilms are these structured things. Every time, you know, you have a biofilm, you have a community, so like inside that has less oxygen, that less permeability to nutrients. So it's like you really have in itself very different environments every time you have a biofilm. Yeah, so structure means physical structure.
So in different communities of plants and animals, there's this concept of the apex species, so that one species controls the concentration, the numbers above it. Does that hold in the bacterial colonies? I mean, do you have one strain that's is an apex strain?
There is now, in fact, what is called the dark matter, you know, in bacteria, because the dark matter, which means we don't know what a lot of this diversity means. If you have a curve, you have like the very, a very, very long tail of things that appear only once in a community. So what does these rare things, mean? So you don't know if 10% for a community means a lot because you don't know what the function is. I mean, you have a single director, it has to be important. Right? You have a lot of people doing other jobs. So we don't know in bacteria, what a keystone species means. There is one paper that I know that tries to simulate that, and just by the frequent occurrence, it tells you, oh, this is a key species. But there isn't enough information to know if that is true, like it is in the macroecology. Yeah, so I wouldn't know. People are more putting attention to things that occur in lower percents, but might have a very important function.
But it's not only the numbers, that the experiment is to delete that one species.
Yes.
And then everything falls apart. Whereas if you delete another species within there, things change a little bit.
Yeah, we want to do that experiment. We really want to do that experiment. We are figuring out how to kill from a complex community just a few, or a few species or something, and see what happens. But I haven't seen that except in modeling.
What you were saying about killing one species sounds like the superbug that everybody is trying to heal. I mean, does any of this work that you do apply to actual illnesses, diseases, bacterial infections?
I think that the only application that that I see is on the knowledge and the concepts, because they are going to be the same. If you go to Patrocinigas and take a plate with the last antibiotic in the drugstore, and then you put all these bacteria to grow there, they all grow on it, okay? So bacteria are the inventors of the antibiotics, and are the inventors of the ways to avoid the antibiotics. So it doesn't matter what antibiotic you go into the wild and test, they all have the ability to avoid it or to kill it or to degrade it. So it's in that way, yes, it is important, because what you learn applies in the gut. Yeah. But then you will eventually have to do the experiments in the gut. Yeah.
I'm curious, typos in humans are Poissonian process. I don't know what about typos in DNA. You have mentioned presence of large percentage of mutants and fast mutation. What algorithm, what technique, what software do you use as your spell checker when you identify mutants? You have to tell a mutant from representative of another species, so you need a spell checker.
Yes, so we isolate each one or a bunch, and we sequence all the DNA. So if it's 4 million bases, we sequence them all, and then we combine them with the wild type bacteria, the one that is not mutated, and we find every change. And it would be almost impossible then to find changes. In bacteria that are just used in the lab, and this bacteria that people use in the lab, like E. coli or whatever, every time you sequence one, you would find new mutations. And maybe those mutations don't mean anything, we don't know. But yes, all these strains are mutating all the time.
So if I could add a nuance to the answer, which I think addresses the point, which is that we only have reference genomes for species that we can culture, right?
No? Well, okay, yes.
Substantially
Now we have the single cell genomics. Now we can sequence, things that we have not cultured, and then we can use that as a reference. But still,
but
most of the knowledge that we have come from these reference strains. Yes. And I think that's going to be a challenge now to figure out if everything is E. coli or bacillus, or we have way more in this diversity.
right, because when you're in a metagenomics environment, single cell genomics becomes a huge statistical problem, right?
Yes. Yes. Yes.
Any other questions? Well, thank you very much for the